I'll be presenting along with Ellen Shashevsky and Kara Higgins. We are Team EPIDEMICS, which stands for Eradicating Pathogens through Immunization Against Diseases Everywhere, using microparticles in controlled release shots. Our team mentor is Dr. John Fisher, and our graduate student is Val Leonard. So to give you a bit of background on the project, the World Health Organization estimates that nearly 2 million people die each year due to vaccine-preventable diseases. This number is largely contributed to by low immunization rates in developing countries. Many factors contribute to low immunization rates, but among them are a lack of proper education about vaccines. Additionally, the long distances that many individuals have to travel in order to get to vaccine sites. So an individual who lives in a rural area or far from a vaccination site needs to travel a pretty far distance to get there, and oftentimes restricts them from getting vaccinated. In addition, vaccination often takes place during work hours, and so work commitments sometimes restrict people from being able to get vaccinated. So when dealing with a multi-dose vaccine, this problem is even worse because an individual has to go 2 or 3 times in order to receive full immunization. And so our goal was to develop a vaccine delivery system that could effectively deliver all 3 doses of a multi-dose vaccine in 1 injection. So we chose to work with the HPV virus, which is human papilloma virus, which is the leading cause of cervical cancer worldwide. 80% of deaths due to cervical cancer occur in developing countries. And this leads back to the problem we previously described because a lot of people can't get to vaccination sites in order to receive full immunization. HPV is composed of virus-like particles, which are the antigenic component of the current vaccines. These virus-like particles are found in the protein codes that are on the outside of the HPV, which is seen in the picture on the left. So our research problems and research questions are centralized around the problem we identified with immunization rates in developing countries. Our research problem is that current HPV vaccines require 3 doses over a period of 6 months, and this often results in low compliance rates and high incidence of disease. Our research questions are twofold. So first is, to what degree can a controlled release drug delivery system induce an immune response? And then secondly, will this immune response be comparable to that of the traditional HPV vaccines? And so we're dealing with cell-mediated immunology, which is describing the ability of cells to identify and then destroy pathogens which enter the system. Specifically, we're working with dendritic cells, and these are antigen-presenting cells that have toll-like receptors on the outside of the cell. And these toll-like receptors have the ability to identify pathogens and different features of pathogens when they enter the system. The dendritic cell is then able to uptake these pathogens and relay this information to T cells, which can come and help destroy them. So as you see in the figure on the left, when a pathogen enters the system, the toll-like receptor identifies the distinct features. It then is able to uptake these pathogens and then bring the T cells in to destroy them. And so we'll be doing this when we make our microparticles. Our dendritic cells will be uptaking this and then using them to create the desired immune response. So the microparticles we've been creating are using a PLGA polymer, and PLGA is poly-D-L-lactic-co-glycolic acid. It's composed of alternating lactic and glycolic acid monomers, and the varying ratios of the different monomers result in different degradation rates. And so on the left of the figure, you're going to see glycolic acid, and the right is lactic acid. So now we'll get into the methodology, and I'll hand you off to Ellen to discuss our specific aims as well as how we're going to accomplish those.
Thanks, John. Our methodology is composed of 3 specific aims. In specific aim 1, we synthesized a collection of unloaded PLGA particles, and we optimized our synthesis technique to obtain particles in the 1 to 10 micron diameter size range. In specific aim 2, we have synthesized PLGA microparticles loaded with ovalbumin, which is a model protein. We plan to present these microparticles to dendritic cells and observe an induced immune response. In specific aim 3, we will be loading PLGA microparticles with the HPV virus-like particles in order to investigate the vaccine-specific applicability of controlled release drug technology. In specific aim 1, we tested a variety of parameter combinations of our synthesis technique in order to yield collections of particles within 1 to 10 microns. And we've identified this as a desirable size range because They are big enough to degrade slowly in the body over an extended period of time, but small enough to be uptaken by dendritic cells. In order to synthesize unloaded PLGA microparticles, we start using PLGA as our organic base, our polymer. We then add this to polyvinyl alcohol, also known as PVA, which acts as a stabilizer. Together, under stirring, this forms a single emulsion, which is composed of PLGA microparticles suspended in the stabilizer. After leaving to stir for anywhere from 7 to 24 hours, we then separate the particles from the suspension using centrifugation and washing. We then resuspend the particles in a buffer. The three parameters that we've been varying in this technique are the concentration of our polymer, PLGA, the concentration of our stabilizer, PVA, and the stirring time. Shown on the left is an image of these microparticles synthesized with this technique under 40 times magnification using optical microscopy. The black scale bar shown is 50 microns, which is a bit smaller than the diameter of a human hair. In this specific trial, we used 3% of our polymer, PLGA, 10% of our stabilizer, PVA, and a 7-hour stir time. This resulted in an average particle size of 8.42 microns, which does fall within our desired 1 to 10 micron range. After we successfully optimized our unloaded microparticle synthesis, we moved on to loading our particles with our model protein OVA, or ovalbumin. Ovalbumin is a protein that comes from chickens that is very commonly used in drug delivery research because it can be used to mimic human virus proteins. It's also very inexpensive, so it's great when we're running numerous trials in our optimization process. In order to synthesize loaded PLGA particles, OVA-loaded particles, we added an extra step to our synthesis technique. We start with our OVA, which we add to our polymer to form our primary emulsion, which we then add to our stabilizer, PVA, which forms a double emulsion. The double emulsion is composed of OVA encapsulated within PLGA microparticles suspended in our stabilizer. We then remove the particles in the same way using centrifugation and washing. We then resuspend the particles in buffer. After we added a step to our synthesis technique, we found that we had to re-optimize our synthesis process in order to obtain particles within our desired size range. For example, in this trial, we used 3% PLGA, 5% PVA, and a 17-hour stir time, and the resulting average particle size was 25.65 microns, which falls outside of our desired size range. In order to address this, we investigated the effects of our separate parameters. For example, in this trial, we investigated the effect of phase concentrations, specifically the concentration of our stabilizer. On the top, we have the same 5% PVA particles that yielded the 25 micron size range, and on the bottom, when we kept the same polymer concentration and the same stir time, but varied PVA concentration, we found that the average particle size was 6.64 microns, which does fall within our desired 1 to 10 micron size range. In order to further characterize and optimize these particles, we will be looking into the loading of these particles. So we will be measuring the amount of OVA encapsulated within the PLGA microparticles. Ultimately, we would like to achieve an optimal loading that will induce a comparable immune response. And this information that we obtained from the loading assays, as well as our size optimization process, will allow us to yield particles that will be suitable for the next steps of our project, which Kara will lead us through.
So after we're able to quantify the differences between our particles, we'll be looking to see what sort of immune response they can induce. So the first test that we'll be doing is the maturation assay on the dendritic cells. So this will measure the ability of the dendritic cells to actually uptake our particles. And this will give us an idea of how likely our particles are to engage in the first step of the immune response mechanism. After that, we'll be using an ELISA, which is an enzyme-linked immunosorbent assay, to quantify the immune response elicited by these particles with the dendritic cells. In specific game 3, we'll be doing the same procedure we did in specific game 2, but instead of using OVA, which was our model protein, we'll be using the actual HPV virus-like particles. So after we are able to optimize the size and loading density of these new HPV particles, we'll be performing the same assays to look at how they interact with the dendritic cells and the immune response they elicit. We will also be testing the normal HPV virus-like particles, so sort of a simplified version of the current vaccine, and this will allow us to compare our vaccine delivery system to the current vaccine delivery system. Upon the completion of Aim 3, we hope to contribute to the body of knowledge about delayed release drug technology and its ability with multi-dose vaccines. So having a 14-person team presents many, many challenges. One challenge that we really faced was scheduling conflicts both with lab and with meeting times. So we just learned to plan ahead and to make compromises with that so that we could all spend enough time on this project. Also with lab equipment and protocols, you have to expect delays with your equipment and setbacks, and you just have to be flexible and work with it. In the lab, even when written protocols are the same, you have to make sure that all members of the team are following those protocols identically. Otherwise, the results could be messed up. And finally, you have to have constant and frequent communication between your sub-teams so that each different team knows what's going on at the same time. So there were a few team dynamics that we put in place to make sure that we could do this project most effectively. One was sub-teams, which I just mentioned. We had a lab team, a data analysis team, and a writing team. We also shared leadership and delegated assignments, so each week we have a different meeting leader, and whenever something needs to be done, we assign it to a different person on the team. We keep a well-detailed lab notebook because when 14 different people are going to the lab, things can get a little confusing. So we always want to know what happened before we went in and when we go. And then communication is really big. We discovered that we needed to have a Facebook group, a group text, and an email just to make sure that everyone was always available and always knew what was going on. So we would like to thank our funding agencies, all of our consultants and collaborators, and all of the groups that have supported us throughout our research and our time on this project. We want to thank you all for taking the time to learn about our project, and now we'd be happy to answer any questions you may have.